Vamos a empezar las conferencias talento del CICA. Hoy está con nosotros Francisco Fernández Trillo, Nano Paco en Twitter. Francisco leyó su tesis doctoral en el año 2004 bajo la dirección de la doctora Susana López Estevez. En el 2006 estuvo en la Universidad de Durham. Estuvo en una estancia con el profesor Niels Cameron. Después, en el año 2008, volvió otra vez a Santiago, porque echaba mucho de menos Santiago, y estuvo durante un par de años en el grupo de Ricardo Riguera, haciendo cosas con Eduardo Mejía, y creo que fue en el 2009, moviste a la Universidad de Nottingham.
Sí.
Estuvo en una estancia postdoctoral estuviste hasta el año 2013.
Hasta el año
2013. En el año 2013 consiguió el puesto de lecturer en la Universidad de Birmingham, y puesto que consiguió, que logró aumentar en el año 2018 como senior lecturer. En el año 2021 fuimos capaces de convencerlo y que se viniese para el CICA. Y pues es un contratado Beatriz Galindo aquí en el CICA desde septiembre, agosto del año 2021. Paco ya lleva muchos años en investigación y ha sido capaz de conseguir casi más de 1 millón de libras, casi más de 1 millón de libras en proyectos, diferentes agencias, tanto europeas como británicas. Como el éxito le acompaña en el tema de pedir proyectos, pues ya ha conseguido un proyecto por el Ministerio recientemente, y se puede decir también, ¿no?
Creo que a partir de hoy sí.
A partir de hoy
ya está público.
Ha conseguido un proyecto de innovación con un proyecto de la CAISA, proyecto de innovación Impulse, bastante competitivo, y bueno, pues algo relacionado con una patente que tiene con la Universidad de Birmingham, va a desarrollarlo en ese proyecto. Bueno, pues Paco, te tocó ahora. Cuéntanos algo, ¿qué es lo que vas a hacer? ¿Qué es lo que estás haciendo con polímeros aquí en el CICA? ¿Qué es lo que has hecho en el pasado? Y bueno, aquí tienes tu audiencia.
Gracias, Jaime. Os voy a contar lo que he hecho, realmente. Entiendo que en inglés. In English.
Jaime, you have to repeat everything.
Everything in English, Jaime. It's fine. Basically, summary very quickly. I'm very old. I've been here for a long time, in Spain, in the UK, and then back in Spain, and then back in the UK, and finally here in Colombia. Okay?
Summarizing, the Morriña is shaking.
Morriña is important, yeah. We live better in Spain. And basically, like I say, I'm going to tell you what I do in my lab, or what we do in my life. To be honest, I sit in an office these days, and people like me actually do all the work. Anyway. As most of you, or a lot of you know, I started as an organic chemist, making small molecules. And through all of those postdocs, I have eventually moved to polymer chemistry. And what we do in the lab is we try to develop tools for biology and medicine. Okay, we try to make things that can be used by biologists, and hopefully with some projects by doctors. Okay, there's a big interest in controlled delivery. Okay, and I'll talk a little bit about this. We started working on antimicrobial polymers. I should say that we now make pro-microbial polymers. We do help bacteria rather than kill it. And I'll talk about this as well. And I'll touch briefly on, we try to develop, or we have developed a method to screen polymers quickly. Okay, and I'll explain a little bit why we do this. So I'll start with talking a little bit about targeted delivery. And this is delivery of antimicrobials, okay? And I don't know if you guys are aware, but antimicrobial resistance is a big issue. And it's going to be one of the leading killers by 2050. Okay, so if you guys look at what has been recommended that we should do to address this problem, there's multiple ways of addressing the problem. The one that caught my attention is this: making better use of existing antimicrobials. Okay, there's a lot of antimicrobials out there, and some of them we don't use them. And the one that caught our attention is this family of antimicrobials, which is antimicrobial peptides. Okay, these are part of the immune system of animals. You can also have bacterial origin. And basically, these are cationic and amphiphilic materials. Okay, so they have high affinity for bacterial membranes. They attach to membranes, they break them apart. Okay, and this is how they work. However, because they bind to membranes and then break them apart, they tend to be quite toxic. Okay, so we don't tend to use them. Okay, now the field of polymer chemistry, I could say I am a polymer chemist now, has really embraced this area. Okay, and there's a million of materials out there. And what we basically try to do is you play with the charge, you play with the hydrophobic residue, you even play with the shape of the material, and you try to maximize activity and minimize toxicity, right? Okay. Medicinal chemistry has a slightly different approach. And for example, with this drug colistin, what we use is a prodrug, okay, where the positive charges are capped, okay, and then in vivo that gets degraded and releases the positive charge, okay. So we thought, okay, let's imitate some of these things. And if these molecules, we started working with Polymyxin B, if the problem for these molecules is that they are positively charged, right, we should be able to put them in front of a negatively charged compound. And if you get things right, these things will come together and form particles, okay? And within these particles, the charge is shielded. You don't see the charge anymore, okay? Now, this is an equilibrium, okay? You have a mixture of mainly particles, but a little bit of the individual components. So this gives you passive delivery, okay? As the system sits at the point of action, you get the slow release of these things, okay? So how did we test it that this works? So we look into our commercially available polymers, and polystyrene sulfonate, you can buy it in different lengths, okay? Polystyrene sulfonate is actually FDA approved to treat metal imbalances. So basically what you have to do is mix these two in different ratios, okay? And you make particles. If you mix them in a 1-to-1 ratio, you neutralize all the charges and your particles are not stable, okay? Nothing keeps them floating in solution, okay? And then precipitate. And you get a flocculate. However, if you get things right, you should be able to make particles. And in our case, we make particles that are roughly 200 nanometers, okay? And because we use an excess of the polystyrene sulfonate, the particles are negatively charged. And like I say, the key message here is very easy to make a library of formulations just by mixing two components in different ratios. Okay. Not only that, you can buy low molecular weight polystyrene sulfonate, high molecular weight polystyrene sulfonate, medium molecular weight. So basically you play with the length of your polystyrene sulfonate, the ratio between the two components, and you have a library of particles. And some of those don't work at all because they're too stable. And basically you have a very slow release. Some of them work and you kill the bacteria, okay? Now we wanted to take this up a notch, okay? Make it a little bit more complicated or make it targeted, okay? We want to deliver the antimicrobial only when there's a microbe, okay? Because otherwise there's bacteria everywhere. And basically what you're doing is you're triggering antimicrobial resistance in your gut, for example, or in your skin. And then this antimicrobial resistance can be transferred to other organisms. So if you want to make this targeted, what we thought we could do is, okay, you get your anionic component, and if this is selectively cleaved by bacteria, basically what you're doing is losing the multivalence in your particle and accelerating the rate release. Okay? So you need to know your enemy if you want to do this, and the bacteria we thought about targeting was Pseudomonas aeruginosa. Okay? This is what people call an opportunistic pathogen. It's everywhere, okay? Normally it doesn't happen anything, but if you have your immune system compromised, for example, if you have cystic fibrosis of a chronic wound, this can be a serious problem. And it's heavily associated with hospital-acquired infections, okay? And you can get really, really serious infections, okay? And the reason why Pseudomonas is so good at colonizing different niches and being pathogenic is because it has a plethora of virulence factors, basically things that they release to overwhelm our immune system and cause an infection, okay? And the one that we thought could be a good thing to target was an elastase. Okay, elastases are designed to degrade proteins and tissues, and this gives the bacteria an advantage. Okay, and if you look at the literature, basically Pseudomonas elastase likes small hydrophobic residues. There's several peptide sequences that it will cleave. Okay, so we thought we did a little bit of peptide chemistry in the past. Okay, it should be relatively straightforward to put those sequences in the middle. We can easily introduce anionic residues by using glutamic acids, for example, and you can even introduce cysteines that if they oxidize, they will cross-link the whole system. Okay. So the first thing we did was try to evaluate which of the sequences work better. You obviously need to get some controls right. The enzyme was working. We knew how much amine was being formed. And looking at these two peptide sequences, what we find is that this GLA sequence is the most active. But more importantly, it's not only the most active, it's completely selective against human leukocyte elastase. Okay, and this is another elastase that is secreted by our immune system as part of the immune defense. Okay, and if there is an infection, you will have human elastase as well. Okay, if there is an inflammation, there is human elastase. So we wanted the system to be selective only for the pathogen and not to be cleaved by our immune system. Okay. There's merit in making systems that are actually cleaved by this, but that's a different story. So having a sequence that was selective for the bacteria, Nacho made a series of peptides with either 1, 2, or 3 glutamic acids on each side of the peptide, and then we even introduced an extra cysteine. Okay, and all these peptides are, most of these peptides are cleaved by the Pseudomonas elastase, and again, most of these peptides are completely selective between Pseudomonas elastase and human elastase. So now we have, as before, a battery of peptides and an antimicrobial that we want to mix. Unfortunately, we couldn't do this with Polymyxin B. Okay, and the reason why we couldn't do this with Polymyxin B is because when you make these particles, the main driving force to make these particles is entropy. Okay, the release of all of these counter ions is really what drives the reaction forward and the formation of the particles. So basically, the bigger the multivalency of your system, the easier it is to make those particles. We had a small anionic peptide and a small antimicrobial peptide. So that doesn't work. Does it make sense? So we had to go back to the literature. We could not use these antimicrobial peptides because they're not long enough. So we tested an antimicrobial polymer. And we were lucky to find that polyethyleneamine is another commodity polymer. You can buy it in different lengths and different sizes, different shapes, sorry. Okay, it's also antimicrobial. It's mainly against gram positive, but it has some activity against gram negative, and actually it worked against Pseudomonas elastase. So, sorry, Pseudomonas. So basically, like I say, you mix, now we have 4 peptides, we can mix it with polyethyleneamine in different ratios. And what you tend to see is that when you put these particles in the point of application, the particles tend to swell. And this is what you see here. Okay. And this is because in the buffer, there's other ions that compete for the electrostatic interactions. They separate the two components a little bit, and the system swells. Okay. In some cases, it swells and then eventually breaks up because the system is too unstable. In other cases, the system swells a little bit, and in other systems, it swells a lot. And actually, this is going to be important for activity. Unfortunately, we couldn't test all of those, but this is where we are. Do these particles get degraded? Yes, they do. If you incubate them with LASB, the Pseudomonas elastase, what you see is that there's a gradual decrease in the number of counts. Okay, and basically, number of counts in DLAS means you have either less particles, the particles are less dense, or they are smaller. And all of those 3 indicate that you are basically cutting your particles. Okay. That doesn't happen just with buffer, and more importantly, as before, if you incubate this with the human elastase, nothing happens. Okay. You can measure also the number of amines in your system, and that's information about cutting. Okay, the peptide generates an amine. Our particles have a lot of amine because we're using an excess of polyethyleneamine in this case. If you treat it with LASB, you see an increase in the number of amines, which again suggests the particles are being cut. And if you incubate it with the human elastase, nothing happens. So basically, the particles are selective as we designed them for the Pseudomonas elastase. And do they work? Do they kill bacteria? Kind of. Okay, if you incubate Pseudomonas elastase with these particles and you look at the activity after 2 hours, sorry, there's barely any activity, 10%. Okay? And this is good because this is what the particles have been designed for, to protect the antimicrobial and keep it from. If you let the system evolve, you recover some of the activity. We were hoping it was here, it was only 20. Okay. So the most important information in this graph is that if you use a bacteria that does not produce the enzyme, you see no activity whatsoever. So basically, in the absence of the enzyme, you have no capacity to cleave this. Unfortunately, the system becomes very stable after 4 hours, and none of the systems we tested worked. Nacho finished his PhD, then the pandemic hit, and life goes on. But on this project, I hope I have convinced you that we've made short peptide sequences that are completely selective to Pseudomonas elastase and will not be triggered by our immune system. It's very easy to make these particles. Just mix the peptide in different ratios with the other components, and you can tailor the size, the stability, and even the charge. We reduce the toxicity, and if you don't have the elastase, you don't give them. Okay. And this is where we are here. Hopefully, we'll get someone else working on this area, and we'll be able to make the system very effective. So what I'll talk for the rest of the presentation is antimicrobial polymers. And actually, they started being antimicrobial, and now, like I say, they are pro-microbials. And this started in a postdoc in Nottingham, where I was working with Cameron Alexander, and we were trying to make polymers to interfere with bacterial communication. Okay. And I don't know how many of you know this, but bacteria talk to each other. There's several ways they talk to each other. One of them is quorum sensing. And the way quorum sensing works is they use small molecules called anti-inducers, okay? And they secrete these molecules. So when the population of bacteria is low, the concentration of those molecules is low and nothing happens, okay? Population of bacteria grows as the bacteria multiply, okay? And so does the concentration of these auto-inducers. And basically, when the concentration reaches a threshold, bacteria detects that and know that there's a lot of them. So they know there's a strength in numbers and they change their behavior. Okay, they say, hey, there's a lot of us now. So it pays off to be naughty. It is basically what they do, right? And the reason why people think that interfering with this communication is useful is because a lot of the phenotypes that they control are related with infection. Okay, they control biofilm formation, and I'll talk a lot about biofilms eventually forming or the expressing of viruses. So the idea is if we stop this communication, they won't tell each other, hey, let's start being effective, and you will stop the infection without killing the bacteria. And that's a main kind of motivation to develop drugs that do not develop resistance, because if you don't kill them, there's less chances they will develop resistance. That's kind of basically the way it works. It's not exactly, as they're very cheeky. Okay. Now, we started from a very kind of academic point of view. So we chose a marine bacteria, Vibrio harveyi. It's not a major issue for us. But there's two things that are really nice about Vibrio harveyi. This quorum sensing network is very well understood, okay? And of the autoinducers it produces, one of them is a Bori tester, okay? And as a chemist, this is a very interesting molecule, okay? The bacteria does not produce the molecule, these molecules, AI2. The bacteria produces the sugar-like molecule, which in the presence of boric acid in seawater, this is a marine bacteria, forms the active compound, okay? And if you guys know anything about boron chemistry, this is a reversible reaction, so we should be able to design polymers that have diols, and then they will compete for the boron, okay? So the boron, instead of being attached here, it will be attached to our polymer, and therefore we will reduce the concentration of the active molecule and make it inactive, okay? Now, the other thing very nice about Vibrio harveyi, it is a bioluminescent bacteria. It produces light. So basically, it gives you a nice readout of things. If they work, what you should be getting is a decrease in the in the amount of light produced, and hopefully you will get a dose response, and you can see whether your materials are active or not. Okay. So with this in mind, Scarlett, that was the PhD student working on the project at the time, prepared 4, well, actually 3 polymers, and we bought polyvinyl alcohol, and we were expecting this to have a very high affinity for boron because it has catechol. Sugars have a medium affinity for boron, okay? And aliphatic diols, like this one, have a low affinity for boron, okay? So we were expecting this is not going to work very well. This is going to be awesome. And actually, science gives you a lot of surprise. This is not exact. This is not at all what we saw, okay? Yes, PVA was behaving as expected, but if you look at this guy, it's not really behaving, right? There's a plateau here. If you look at the sugar polymer, it seems to go up and then it works really well. And actually one of our polymers was actually helping the bacteria produce more light. So if this was a therapy, it will be terrible, right? We're making bacteria be more active, right? And actually this really depends on when you look at the data. This is 5 hours. If you look at the data at other points, some of these polymers are actually increasing light production, okay? So what the hell was going on? We were terrible at making polymers. No, we're reasonably good at that. It took us a while, but what happens here is all these polymers aggregate bacteria, not PBA, but all these 3 aggregate bacteria. I told you before, cationic charges like negatively charges on the surface of bacteria, electrostatic interactions, we use those to make particles. You can make aggregates of bacteria. We don't really know how the catechol works, but some bacteria have receptors to capture iron, as some of you may know. Okay, so it may be that. Catechols are really funny because they oxidize and they form polymers, right? So it could be that they polymerize around the bacteria and they trap them. What I can tell you is that there is an aggregate of bacteria there, okay? And then Vibrio has a lectin that binds to glucose. So basically these 3 cluster bacteria. And what we were able to show with the help of a computational scientist is that what is happening here is you have bacteria and the signals, and the polymer basically brings everything together, okay? And it forms a cluster where the bacteria are trapped, okay? And within that cluster, there's very low diffusion of the signal outside, okay? This is a very dense as compared to your buffer, right? This is very dense. So basically the signal does not diffuse. So basically you're fooling the bacteria and they're thinking, you know, there's a lot of us. Actually in that spot, there's a lot of them, okay? So basically we've told the bacteria, okay, start being naughty. In this case, start producing light, okay? And the way we show this works this way is we just basically made a polymer that has no way of interacting with the signal, no diol to bind to the boron. It just aggregates bacteria, okay? And you can see that, for example, if you measure the size of the clusters, okay? Bacteria is roughly 1 micron, and we were making 20 micron clusters, okay? And the polymer, no matter what the concentration we tried, in all cases, enhances the production of light, okay? And the way it does it, Louis was the computational science on the project. He was able to model this and reproduce the behavior. And the way it does it is, like I say, it brings the bacteria together. The bacteria thinks there's a lot of them, and it starts producing light, okay? So is this useful at all? Can we use this to treat bacteria? That was my motivation when I moved to Birmingham. And I met a great collaborator, Marie Prackler, that was working on cholera. And while cholera is not an issue here, it's kind of a major issue in developing countries, especially if there's poor sanitation, right? And it still causes a lot of deaths worldwide. Okay? The way cholera works is you drink contaminated water with cholera. It will colonize your gut. It will stick to your gut. And basically will switch phenotypes, produce a colony, produce the toxin. The toxin gives you diarrhea because they like seawater, okay? And if you don't drink and rehydrate, you die of dehydration, okay? Now, at some point in this cycle, bacteria activates quorum sensing. They know there's a lot of them. This is not a nice neighborhood anymore. They switch off the production of toxin. They switch off the production of the tea light that makes the colony, and they move to a new place. And this is terrible because if there's no good sanitation, all this bacteria goes into the water and we drink it out, so on. So what we thought is, okay, our polymers should be able to bind bacteria and maybe they prevent the adhesion to the gut, okay? But also, if they activate quorum sensing, they switch off the production of the toxin, okay? So it's kind of a win-win situation, right? We either stop them from binding to the gut and we stop them from getting into the, from producing the toxin. So Nico was the person that started this, and it's reasonably easy to make polymers again that bind to bacteria. We continue with the cationic charges. That was the easiest way of doing it, okay? Nico prepared 2 polymers. He hated making polymers, but I was able to make 2, right? And basically what you're seeing here, we had a fancy microscope, are those clusters of bacteria, okay? Now, very importantly, cholera is quite tough. So with this polymer, it was viable at all the concentration, it was growing at all the concentration we tested. This polymer was a little bit toxic, but actually, if you look at the bacteria here, almost 90% of them are viable, okay? We still had a concentration where we can work, okay? So we were not killing the bacteria. Again, that's important for us. We don't want to kill bacteria at this stage. We want to stop quorum sensing. So how are bacteria responding? And if I remember correctly, when I started showing some of this data to a microbiologist and I was telling we're bringing bacteria together and making clusters and they activate quorum sensing, he said, Paco, you're just making biofilms. And this is what bacteria do apparently, right? So what? Like, okay, let's look if we're making biofilms. And indeed we are. You can look at the amount of biomass that the bacteria produce using crystal violet. And you see that the polymer increases the amount of biomass. If you look at DNA, which Cholera uses extracellular DNA in their biofilms, you can see that in the absence of polymer, all the DNA is punctuated, which is mainly located inside the bacteria. If you add the polymer, there's a lot of DNA outside the bacteria. And actually, Nico loved making reporters, so he was able to make a couple of reporters of the genes that regulate the proteins that regulate the biofilm. Okay, RBMA is one of those. It's responsible for bringing bacteria together. And we were very happy to see that in the presence of our polymer, this goes up. Okay, so the polymers are activating biofilm formation in this bacteria. And I'll come back why we are happy with this. Ollie was very keen on doing microscopy, so he was able to take kind of videos of these bacteria. And again, in the absence of the polymer, bacteria just grow, spotted. There's not a very good resolution, but I hope you trust me when I say, these are just kind of mainly individual bacteria. If you look at what the polymer does, you will see that, very quickly, they start growing in biofilms, okay? And it is actually, it's like growing a crystal for those of you that do crystal, right? Basically, the polymers nucleate and you get a nice biofilm. And if you look at the literature, Bonnie Basler has been able to capture recently one of these with high resolution microscopy. It's not one of ours, it's one of hers, okay? But this is Cholera biofilm. And, I hope you can see that some of these really resemble, right? Again, the polymer is basically telling the bacteria, go and make a biofilm. So is this a good biofilm, which is basically what cholera will be on the environment, is in the sea, it's not killing anyone, it's just sticking to things, right? Or is this a kind of virulent biofilm or aggregate, right? Which is what you get in the gut, right? So again, Nico made a series of reporters, including the main regulator of toxicity, and the regulator of the cholera toxin, and in the presence of our polymers, those get downregulated. So basically, the polymer is telling the bacteria to make biofilms, but those are good biofilms. They're not toxic biofilms. Okay. Does this work at all? Kind of. We were able to kind of see if the polymers were able to inhibit colonization of the gut in an animal. Several fish is a very nice animal because it's transparent. And apparently it's not an animal if you kill it before X amount of time. So it's awesome. And we were happy to see that, we could reduce the amount of bacteria in the animal. Basically, this is what happened. If you put the polymer in solution and then you add the bacteria to the fish, right? If there's no polymer, the gut gets colonized. On its own, it has nothing, right? And the polymer basically reduces, and you can count the number of bacteria. So it works really well. The main problem is cationic polymers are shit and they're going to kill everyone. So, it was just proving that the idea works. Sorry, no, that doesn't work. Okay. So at this point, we're saying, okay, polymers are really nice. They seem to work and inhibit cholera toxin. We seem to be increasing biofilms. I don't know if this is good or bad, right? Now, is this selective? Like I say, cationic polymers are not selective. They're mucoadhesive. They bind to everything, right? So we should be making things selective, right, and non-toxic, because actually those polymers are quite toxic to the fish. But what I started kind of thinking is there any chemical signaling here? Is chemistry telling the bacteria to do things, or is it just mechanical chemistry? It's just the aggregation. What I want to explore here, and this is something we started on recently, is okay, can we design a library of polymers with different chemistries so that when I incubate these polymers with bacteria, I activate something. So maybe I make the bacteria more resistant, right? Biofilms are basically that. Biofilms make the bacteria more resistant, right? Or we could make the bacteria produce something, right? It starts producing a pigment, and maybe the pigment is useful. And maybe polymer 3 does both things, right? So can we start playing with polymer chemistry to tell bacteria what to do? Okay. Now, if you want to do this, you jump into a kind of a drug discovery or material discovery problem. Okay. And with polymers, there's two problems. Okay. The first one is hard to get structure-activity relationships. Okay. If you get a small molecule and you change something in your small molecule, And I know some of you have been in Granada and saw some of these already, so bear with me. If you compare small molecules and you see molecule 2 is more active, you can start saying, okay, it's because I introduced this functional group here, I'm going to explore that space, right? In polymers, it may not be the case. Polymer 2 may be longer, and you don't know whether it's the length or it's the chemistry, okay? How do you solve this? Post-polymerisation modification, okay? You get a reactive polymer, and you can modify it. I call these modulators. So basically, you can make libraries of polymers that will have the same number of units if you get the chemistry right, okay? So now you can infer this works better because it has 2 browns and 1 blue, okay? Then brown and blue are good. And you ignore the length problem. Now I'm quite lazy, so this is actually still a huge problem because most of these materials don't work. This is drug discovery 101, this is material discovery 101. You make a material, a drug, it doesn't work, chuck it down the sink, right? So basically you're investing a lot of time purifying materials that are not going to work. So can we do this in situ? This was our next question. Can we do the post-polymerization chemistry in water or as close to water as possible? So basically we can then add our biological, right? We can screen for activity without purifying and isolating these guys. We see what works, and then we refine, okay? And I don't waste time purifying things that are not going to work, okay? If you want to implement something like this, you need to know your click chemistries or your high efficient chemistries. That was all the hype in 2008. And basically, a lot of these have issues for us, right? They're either not soluble in water, acids are not very soluble in water, alkynes, right? Another had sign reactions, right? Thiols, as I told you, oxidize, activators, esters hydrolyze, okay? So the chemistry that caught our attention is the condensation of an amine and an aldehyde. This is heavily used in bioconjugation, right? You can get different amines, and basically they will form this faster or slower, but more importantly for us, the bond will be more or less stable. Thank you for coming to my talk. So if you use an imine, this is a very dynamic reaction. It's very unstable, good probably for drug delivery. But if you form an oxime, that's very stable, okay? And acyl hydrozones are somewhere in between, okay? And for those of you that were interested in click chemistry at some point, this is click chemistry, okay? It's not me, Charles said that in 2001 when he coined the term, okay? Caroline Bertozzi had already explored this chemistry to make libraries of glycopolymers. So we thought, okay, this is the way to go. The reactive polymer is going to be very soluble in water, okay? All those hydroxides, because they make a lot of hydrogen bonds, they are very soluble in water. And if you go to Aldrich's catalog, there's a million aldehydes. Some of them we have to make, but others we just found, okay? You can do this mainly in water, okay? Sometimes we have to add a little bit of organic solvent, but often this gets diluted into your assay, and then you can basically. So where did we start it? Actually, I started this work with Javi Montenegro, who did his PhD with me, and we're very good friends. So we make that polyhydroside, and we explore nucleic acid delivery. So basically here, what you need is a cationic group, and we fix the cationic group because apparently this works really well, vanadium, and we explore the hydrophobic space. And very quickly, you can identify that, for example, for lipidic vesicles, model membranes, aromatic groups are very good. But if you go into cells, actually, these aliphatics are a little bit better. If you change your nucleic acid and you use plasmid DNA, you have to use a longer polymer. Okay? And actually, our system is very modular. In one assay, we can tailor or we can test the length of the polymer and the composition. And we were able to find one formulation that worked really well for plasmid DNA. And eventually, we tested also for messenger RNA. The polymer had to be even longer. Messenger RNA breaks as soon as you look at it. And we had to use very long lipids, okay? So how can we use this for bacteria? How can we start playing with bacteria? Or how did we start playing with bacteria? So Nico actually started a little bit. He showed that he was able to use the chemistry. If you put an imidazole, and you do the chemistry at acidic pH, this is cationic. It clusters bacteria. It reduces the toxin. It reduces the toxin co-regulated pH, okay? And again, cholera is quite tough. Only at the highest concentration, it was not growing very well. Ollie decided to make a little bit twist and play with different ratios of these two. And basically, the motivation here is, okay, imidazole should be more hydrophobic than our hydroside. So basically, I'm playing with hydrophobicity. As I increase the amount of imidazole, the polymer becomes more hydrophobic, and actually it forms more biofilms. And in this case, both the biofilm and the toxin were going up. Okay, and we think this is heavily associated with the toxicity of the polymer. Actually, the polyhydroside is toxic, and the polyimidazole is toxic, and as you increase the concentration of the imidazole, the polyhydroside is toxic, and as you increase the concentration, you reduce the toxicity. And 75% seems to be a good balance between stressing the bacteria a little bit. If you stress this, if you poke people on the eye, they'll start moving very quickly, right? But not killing them. And that was basically where we are. So with Tim Overton, we started playing with E. coli, and this had a motivation to use this in biocatalysis. So we tested mildly cationic polymers and aromatic polymers, and it seems that for E. coli, aromatic aldehydes are the way to go. And actually, the way we show that this is the case is we change the media. All these cationic polymers became neutral. And actually, what you see is now all of them are hydrophobic, and we really increase the formation of biofilms. Okay, so it seems that E. coli likes hydrophobic aldehydes and hydrophobic polymers. Okay, and basically, this is a picture of the aggregates at low pH with this polymer, and then when we change the pH and we make it neutral, we get all of that. And like I say, it seems that E. coli likes aromatic residues, anthracene, naphthalene, all of those work really well. Heteroaromatics like pyridinium don't work as well, and aliphatics don't work very well for these microorganisms. If we use a different polymer with a different shape, and this is very preliminary data, actually now the aromatics don't work really well. And it seems that the heteroaromatics are better. And we also tested some cationic, anionic, and some kind of fancy. And what is very interesting is this depends a lot on the bacteria you're using. Okay, so for example, Pseudomonas fluorescens makes good biofilms already. It's a good biofilm former. So if you look at the numbers, we're not really helping it much, right? It's already there. It's already good at making biofilms. But Bacillus and E. coli are not that good. And actually, we're helping them. The numbers are bigger. Right? What is also very interesting is for Bacillus, the best polymer was a cationic one. While for Pseudomonas, it seems that it likes aliphatic residues. And basically, this is telling you that not all bacteria are the same, right? They have really big difference on their surfaces, and they like really different things. Okay. And I guess what I'm trying to say here is we can screen for different things very quickly, right? If you don't know what your bacteria likes and you want to aggregate bacteria, we can very quickly screen and try to identify whether it likes, an aliphatic one, a cationic, or a heteroaromatic. Okay. And this is not only crystal violet. With coli, we look at curly, which is a protein and adhesin that helps the biofilm stick to things, right? And again, you see that the adhesin is upregulated. Interestingly, now aliphatic work really well. And we think this is because curly is overexpressed before the biofilm settles, okay, when the bacteria start aggregating. Okay, so for some of these, it's already settled and it's not as needed. And actually, what I was all the time in my head, what I was trying to do is, okay, can we make a bad biofilm former make good biofilms, right? And I'll come back why you want to do this. And normally, what you may do this by gene editing, okay? You'll introduce a gene or you'll mutate a gene, right, into the bacteria, and it will start producing biofilms, okay? Again, our question is, can we do the same but using a synthetic polymer, okay? So for this, you need to have two strains. One is a bad biofilm former, and one is a good biofilm former. And these two are E. coli. As far as I know, these are isogenic strains. There's only one single point mutation that makes the second one a good biofilm former. Okay, so it's the same background, different phenotype. And the whole idea is if I add my polymer, the bad biofilm former starts making good biofilms. Okay, so now I can use it as a strain if I think biofilms are good, and I come back and I'll tell you the biofilms are good. You have to be careful because if the good biofilm former falls on the pot, you can even make a better biofilm. Okay. And you need your controls. And again, this is not only curly, which is the adhesin, it's also crystal violet. Okay. We've explored whether this is reversible, and the chemist in the audience will realize that the chemistry we're using is reversible. So actually, you should be able to change the conditions of your culture and revert the biofilm. And this is very preliminary data. Unfortunately, we haven't been able to explore this further, but we're very happy to see that we can switch off the biofilm just by changing the culture media. And we can actually reactivate it, okay? And I think this is probably very cool. You know, trying to kind of switch on and off how bacteria stick or not to surfaces may be very useful. So what's the point? Why are we making biofilms? Most of the time I get, Paco, you're mental. Why are you making biofilms? Well, actually, biofilms are very useful. Okay, they're quite important in human and animal health. They're part of our gut and keep us healthy. Agriculture, that's quite interesting, coating seeds with bacteria. They coat plants. Okay, they have been heavily used in bioremediation and waste treatment. All those flocks on clean water are basically bacteria. Okay, they're part of our food production. All your cheeses and vinegars come thanks to biofilms. And Tim was very interested in biocatalysis. Okay, he wanted to use this for biocatalysis. And basically what you're doing here is two things. You're taking advantage of the intrinsic resilience of the biofilm. So bacteria produce biofilms to protect themselves from the environment. So basically in a biofilm, you will be able to treat bacteria harsher. Okay, so maybe you can do chemistry that you will not be able to do otherwise. Okay, or biocatalysis. And then this is a self-contained system to a certain extent. You have to feed it, but then, you know, the bacteria is responsible for recycling the enzyme, the cofactors, and actually cofactors tend to be a big issue when you do single enzyme biocatalysis. Okay, Tim was very interested in this, and the reaction we explored was the transformation of a haloindole into a halotryptophan. Okay, and again, the bad biofilm former was terrible at doing this. The good biofilm former was really good at doing this. And when we add our polymers, the bad biofilm former starts to behave like the good biofilm former. Okay, so we're helping the bad biofilm former. And actually, you have to look not only at the formation of tryptophan. This is a reversible system in the bacteria. The bacteria produces naturally an enzyme that cleaves here. So normally you will look at the conversion of indole to tryptophan. And again, the good biofilm former is better than the bad biofilm former. And our polymers make the bad biofilm former behave better than the good biofilm former. So we think this is pretty cool. So I hope I have convinced you that actually, you know, synthetic polymers can start talking to bacteria to a certain extent, that you can get some useful applications out of this. Okay, you can make polymers that increase the biocatalytic activity of a biofilm. And again, if you want to screen polymers for biological activity, polyhydrosides may be the way to go. We can do gene delivery, we can do bacteria. Okay. Don't know how long I've taken, but I'm going to stop here. I've actually told about everything we do in my lab. I just need to thank the people that did the work. Like I say, I sit in an office. I sit everywhere with a computer. That's basically my office. The collaborators, the people that pay the bill, in particular, Ministerio de Educación y Ciencia, that has allowed me to come back to Spain and, you know, enjoy Morriña. Thank you.
So it's time for questions. So
Happy to take questions in Spanish, in English. Questions I leave.
Yeah, I have a question. Yeah.
Thank you, Alex. Thank you. I'll pay you.
It's just a curiosity.
Yes.
Could you apply your polymers for coating surfaces? Because I think it's a very good application if you are having some problems with biotoxicity. Yeah.
So we wanted to do that on the E. coli project. So with Tim, we had a student. He was a biotechnologist. He was from Jerez and had been working in wine production, obviously. So we said, oh, we're going to coat surfaces. But like I say, he was a biotechnologist. I'm a polymer chemist or a synthetic chemist. And Tim is a microbiologist. So we were terrible at surface stuff. So halfway through the project, we said, we don't do this. We do what we know. And we use the polymers as additives. And I think there's merit in adding the polymers as an additive, right?
Yeah.
It can be kind of a variation of whatever your culture media is, right? But I think there's a lot of merit in making kind of surfaces that control, and kind of rewritable surfaces and stuff like that. That's very cool. Do you know how to work with surfaces?
Yeah, maybe combining your polymers with 2D materials. That is of course my field. It could be interesting.
Yeah.
Yes.
Yes.
So we can talk. Yeah.
I'll pay you a beer for asking a question. This was all a stage, okay?
And another question. Yeah.
Okay, thank you, Alex. 2 beers.
Yeah, when you are activating in the last part that you are reactivating your biofilm,
Yes.
your polymer, this activation is higher
Yeah.
than the
Yes.
first one. And if you are using this kind of chemistry, it's quite surprising, right? At least
We don't know why. So we were very happy with this. This is like awesome, right?
Yes.
This is like, good biofilm, switches off, same level, right? Because there's I don't remember, I think there's between 6 and 12 hours between these 2 points, right? You have to let, biofilms are slow growing, okay? Most of the data I show is either 24 hours, 48 hours, and for some bacteria, you have to grow them 72 hours. So there's obviously a growth in here, right? So you can see that your control is also making more biofilm because you just let it longer, right? Let it grow for longer. So I guess this is expected as well, right? That it goes and it grows for longer, right? Again, we were surprised that it was so good because be aware that we only add a little bit of polymer. The bacteria then produces all their own kind of biofilm. So why it's so effective at switching off the biofilm, we don't really know. Maybe the microbiologists in the area can help us.
I think maybe it could be related with the reproduction of the bacteria, because from a biological point of view, if you are breaking and restarting the biofilm, but the compounds are in the media, so it's easier to grow more at the beginning.
Yeah.
Because they started to reproduce. So is that possibly a biological explanation?
Yeah.
Could be.
I mean, they do have their system to kind of eat the biofilm away, right? Because that's what they'll do. At some point, they'll eat a little bit of biofilm and move. That's what cholera does. So something like this has to be involved in this case. And maybe it's toxicity. Remember, the backbone is a little bit toxic for bacteria. So maybe at this point, we just kill them a little bit and they say, hey, let's leave this place, right? But it's very, again, from my perspective, it's very interesting, right? You switch it on, you switch it off.
So Paco, what part of the presentation you made is going to be part of your national, the project that you just
So it's the
got
gene
from-
delivery one. So the gene delivery is the one that I'm working with Anna. So we are continuing with this. So this was all done with either HeLa cell or I think it was CHO, I think it was. So it's all immortalized cells. So Anna has convinced me that there's merit in doing gene delivery to mesenchymal cells, stem cells, MSCs, way to go. So this is basically what we're trying. And again, it's the same idea. Can we quickly identify something that works? Because these are very fussy cells, right? They will die easily. They're hard to transfect. Right?
And the other part of the Impulse project from the CICA is the part that you
It's also following on this.
Yeah.
So we started doing delivery to the eye. Unfortunately, that's not protected, so I cannot talk about the chemistry yet.
Okay.
But it's around the same space. It's drug delivery. We're going to explore nucleic acids, but also conventional drugs to deliver them across the eye because the eye is designed to not take things. It's basically how it works. But there's a million eye diseases, right? So apparently, La Caixa like that. And I now go every 2 weeks to Barcelona. And they train, actually is an awesome. So if you have anything that is a little bit translational, consider going because they train you and it helps you a lot to frame. It's very different how we talk to academics and how you have to talk to businesses, right? So it's a good training to know how, you have 1 minute, convince me, give me the money, right? So I would encourage, and again, if anyone needs help on putting one of those together.
I would ask.
Yeah, sure. Sure. I'm hoping to continue the other projects as well. I think the biofilm if anything, is fascinating, right? Bacteria are amazing, right? They just adapt to everything and they eat everything and they grow anywhere, right? You know, it doesn't really matter if it's sulfur, phosphorus, or whatever. They just do it, right? So we should try to harvest them rather than just kill them. I mean, we have to kill the bad ones, but you know, if we can harvest them, and I know there's group here that do this, right? So I think that's fascinating. And then the drug delivery it's again, antimicrobial is, you know, it's interesting. It's very interesting.
Okay, so there is no more questions. So let's thank Paco for this nice talk, and
Thanks.
we wish you the best for both projects. So, okay, so thanks a lot for the amazing talk.
Si es que entraba ahí al final, un poco bien.